Good day everyone, welcome to the course on next generation sequencing technologies, data analysis and applications. In the last class we started discussing the interpretation of DG analysis results, and as part of that we have talked about functional enrichment analysis. This is a very common analysis that we do to understand the functions of the differentially expressed genes, okay. And what you want to know is that whether there are specific functional classes that are up regulated or down regulated, right, in certain samples, right, and this will give us molecular insights, right, and insights into the biological processes that are happening inside these cells. Okay. So, this is what we are going to discuss in this class, right. So, we'll be talking about this functional enrichment analysis, the actual enrichment analysis part, okay, and this will be the agenda for this class. These are the keywords that will come across. One is hypergeometric test, the other is GSEA. Okay. So, we will see this is the gene set enrichment analysis. This is a specific tool that actually performs this enrichment analysis. Okay. So, just to remind you, right, we do something called functional enrichment analysis. This is also sometimes called gene set enrichment analysis or pathway enrichment analysis. They are not exactly same, but although used interchangeably. So, what functional enrichment analysis tries to do is to answer these questions, right. So, whether there are specific functional classes that are over or under represented in the list of differentially expressed genes, and as I mentioned, the ultimate goal is to understand whether these pathways play any role in disease progression if we are doing this disease versus healthy sample comparison, or whether they have any role in response to therapy, right. For many diseases some therapy does not work, and in that case we want to know, okay, which pathways are actually responsible for this therapy resistance and so on. In case of let us say we are analyzing stress response studies, right, where we expose cells to different conditions, right, condition 1 versus condition 2 and so on. We want to know which pathways are actually helping in stress adaptation, right, in cells or in organisms. Okay. So, these are the ultimate goal of the analysis, and this actually give us more insights into the biological process and help us understand the molecular basis. So, for doing this functional enrichment analysis, the first step is to define the gene sets, okay, and this actually associates genes to specific functions. And in the last class we talked about different resources where we can find this information, right. So, we have introduced gene ontology or GEO, where you have the ontology part and the annotation part. So, the annotation part is organism specific, where you are associating genes in organisms to specific geoterms and hence to specific functions. Then you have talked about the cake database very briefly. So, this is again associating genes to molecular pathways or biological pathways, and then you have the MSigDB database, which connects genes to some gene sets, right. So, it organizes genes into gene sets, and this could be signature gene sets or hallmark gene sets. So, once you have these gene sets, you can now do the enrichment analysis. Okay. So, there are two different approaches statistically for doing this enrichment analysis. The first is actually the enrichment part, okay, and the other is overrepresentation analysis, and we will discuss these two methods, okay, how we actually go about this enrichment versus the overrepresentation. Okay. They are statistically different. So, we will go into details about these statistics. Okay. So, the methods for enrichment analysis, they actually test for enrichment of functional classes within a list of gene. Okay. So, in our scenario, right, where we have done the differential expression analysis, this list of genes will be the genes that are differentially expressed, right. So, these methods actually test whether certain functional classes are enriched within that list, okay, and they usually compare this based on expectation from a global gene list, right. So, if you are looking at an organismal level, right. So, this would be the global gene list, or maybe the gene list that you started your experiment with, right. So, maybe you are studying only a part of the genome, right. So, that will be your global gene list. Now, these methods rely on specific statistical tests. So, some of these methods are implemented in binomial tests and some of them are implemented in hypergeometric test. Okay. Again, we will talk about these differences when you go into the actual statistical test. So, here are some tools, of course, there are many more out there, and the links, right. So, some of these tools are available online. So, you can simply paste your list, you can give the reference list or the global list, and you can compare whether there is any enrichment, right. So, you can find the results through these online tools. So, we will talk about now this binomial hypergeometric test based method and how it works, right. So, what is the idea behind this hypergeometric test or binomial test based method? So, we will take an analogy first and then we will come back to the gene analysis, right. So, let us imagine, right, we have a box and we have 100 balls in there, okay, and out of them you have 20 red, 25 blue, 30 green, 15 orange and 5 black. Okay. So, what we do, let us say out of this 100 we pick out 20. Okay. So, we pick out 20 balls in another box, and what we see is that out of these 20, 4 are red, 1 is blue, 12 are green and 2 are orange. Okay. Now, the question that we ask, for example, is that is this distribution of colors, right, in this sample expected by chance alone, right. So, if you randomly pick these 100 balls, would you get this distribution? Is this possible? Okay. Or is there an enrichment for a specific color, right, or do you see this is not a random sampling, maybe we have picked out specific colors preferentially. Okay. So, once you think of this example, this actually is very similar to what we want to do in case of hypergeometric test based method or enrichment. Okay. So, this is the global box, and in our case this would be the global gene list. Okay. So, where we have about 10,000 genes, and let us say instead of the colors here we have now these functions. Okay. So, we have these x1 functions, x2, these are the numbers, right. So, we have x1 genes that are in function class 1, x2 number of genes in function class 2, xm number of genes in the function class m, right. So, this is the distribution in these 10,000 genes. Now, let us say we have got this list of differentially expressed genes, and in our case let us say we can imagine we have got 100 genes, right, in the list, and in the 100 out of these 100 genes we see y1 number of genes that belong to function class 1, we have y2 number of genes that belong to function class 2, and we have ym genes that belong to function class m. Okay. Now, once we have seen this, right, the question is there an enrichment for a specific function, right, or is it just a random sample that we will get, right. So, the question is would we get the same distribution, right, this distribution y1, y2, ym if we just pick a random sample from this global gene list. Okay. So, this is actually what we are going to answer, right. So, this will tell us whether there is an enrichment in the list, right. Now, the decision on whether we want to use binomial test or hypergeometric test. So, binomial is sampling with replacement, if you go back to your statistics classes, and hypergeometric is sampling without replacement, right. So, what you see in our scenario, this is mostly sampling without replacement, right. So, if we pick a gene from the full list, that gene is not replaced, right, we cannot pick that gene again. So, this is a sampling without replacement in our case. So, we should use the hypergeometric test, and this is the most appropriate out of these two. Okay. But some of the tests, some of the tools still use binomial test, and this is an approximation of the hypergeometric scenario. Okay. So, what we do in this case, right. So, the question is whether there is any enrichment for specific function in the list of differentially expressed genes, right. So, on the right hand side we have the list of differentially expressed genes. Now, to answer this question, what we do is we perform hypergeometric test, and this is done on all functional classes, right, one by one. Okay. So, we start with functional class 1, then go to functional class 2 and so on, and we continue this up to functional class m. Okay. And we do hypergeometric test for all these functional classes. Okay. So, we start with functional class 1, right, and we will discuss, right, how we do this hypergeometric test. Okay. Now, for functional class 1 we have x1 genes, right, that belong to this functional class 1 out of 10,000. Okay. So, what is the proportion, right, of these genes that are in functional class 1 in the global list, this is x1 out of 10,000. Okay. Now, what given this proportion, if you pick these 100 genes randomly from this global list, how many genes would you expect in the sample to belong to functional class 1, right. So, the expected number would be, if it is a random sample, this will be functional x1 divided by 10,000 into 100, right. This many genes we will expect in this sample to belong to functional class 1. Now, based on this expectation, now what we want to do is we want to now compare this y1 with this expectation, and we want to ask, right, how different or extreme is y1 compared to this expected number x1 divided by 1000 into 100, right. So, this is the statistical test that we want to do. Okay. And for that, as we have said, right, hypergeometric test or hypergeometric distribution is the best method, right. And we can transform this question into this question, right, when we are going to distribution based test, is that how often can we observe a more extreme value compared to y1, right. So, how common is y1 or how extreme y1 is, right, based on the expectation that we have set. Now, if y1 is greater than, right, this x1 divided by 10,000 into 100, this is the expected number, we have more than expected by chance, right. So, if we pick a random sample, right, this would be the number, this is the number that would be expected, right. So, this is what is expected by chance. If y1 is greater than that, we will say we have more than expected by chance. On the other hand, if y1 is less than this expected by chance, right, so we will say we have less than expectation, right. So, this is the expected again, and if we have less than that, we say we have less than expected by chance. But now the question is, right, whether these differences are significant, right. We have not talked about anything about statistical significance here, right. We just said, okay, this is more than expected by chance or less than expected by chance, but we have not talked about significance. We need to calculate the statistical significance, right. Without that, we cannot have any decision, right. So, let us look at the statistical test that we can do, right, and maybe we can calculate a p value, right, which will help us make this decision. Okay, so, we have mentioned, right, x1 follows hypergeometric. So, x1 is the number of cases, right, that number of cases or number of genes that belong to this function class 1, right, in the sample. Okay, so, this is the variable that defines that. Okay, and this follows hypergeometric with these 3 parameters, capital N, this is the number of genes in the global list, right. So, this if you are doing this genomoid comparison, you will have all the genes of the organism in this list. So, you just simply count that number. X1 is the number of genes that belong to function class 1 in the global list, and small n, this is the number of samples or number of genes in the sample, right. So, number of genes is 100, for example, in our scenario, and this small n would be 100. What we calculate is this, right. So, what is the probability that this x1 is greater than y1, or the chance of observing more extreme values compared to y1. Okay. So, this is the probability that gives this probability value, right. So, this is what we calculate, okay, and this is given by the hypergeometric distribution. So, if you look at the hypergeometric distribution formula, this comes from this hypergeometric distribution, and we are taking the sum, right, because we are calculating this probability of greater than y1, x1 greater than y1, okay, and this is calculated by k equals to y1. So, the sum is over k equals to y1 to minimum of x1 and small n. Okay. So, this will give us the probability of observing more extreme values compared to y1. Okay. So, you can look at the hypergeometric distribution formula and you will see we can calculate this, okay, in this way. Now, once we calculate this, right. So, here just to mention, k is the number of genes associated with function 1, right. This is what we have discussed. So, this can vary from y1 to x1 or n, and then you have capital N, this is the number of genes in the global list. So, in our example, this is 10,000 and n is the number of differentially expressed genes, that is 100, okay, in our example. Okay. So, this probability value, this probability will give us the p value, right. So, if you have, let us say this comes to 0.01, right. So, we very rarely will see more extreme values than y1, right. So, that will give us the p value. Similarly, if we can think about the other situation, right. So, if y1 is less than the expected value, we have less than expected by chance, but we need the statistical test or the p value from this. Again, x1 follows this hypergeometric distribution with these 3 parameters, and here we calculate this probability of observing more lower values than y1, right. So, again we are looking for extremes, right. So, how extreme y1 is in the whole distribution. Again, the formula is this part is same, we are taking the sum differently, right. So, this starts from 0 to y1, right. So, we can see let us say 0 observations, or we can have up to y1, right. So, we are again looking at how many extreme values, or how likely it is to observe more extreme values compared to y1, and the k n, capital N and small n, they are same in this case. Okay, so, once we have this, right. So, once we calculate this probability value, these sums, right. So, for these equations we know all these values. So, we can calculate the probability. Okay. We know x1, we know k, right, we know small n, we know capital N, right, depending on a situation all these values we know, and then we can calculate these probabilities, right. So, these probability values, they provide p values for these tests, and after we have got these p values, we have to do the multiple hypothesis testing correction, because we repeat this test for all functional classes, right. So, or gene sets, right. So, if we have let us say 8, so maybe 100 functional classes, we are doing this hypothesis test or hypergeometric test for all functional classes, right. So, we are doing 100 tests. So, we would have to do the multiple hypothesis testing correction we just talked about, and usually we use FDR correction, right. So, it is again we can tolerate certain false positives, maybe 5-10%. So, it is okay, right, we use this FDR correction. So, again we set this FDR less than 0.05 or 0.1 for identifying significantly enriched or underrepresented functional classes in the list of differentially expressed genes. Okay. So, what are the drawbacks of this method? This method is quite simple, right. We are taking the list and we are looking for the enrichment, right, compared to the global expectation, right. So, global expectation means if we look at all genes in the organism. So, what is the drawback, or what are the drawbacks of this method? So, this enrichment that you will get is dependent on the gene list that you are providing, right. So, if we have let us say 100 genes, we will get some sort of enrichment. If we have let us say 120, 150, we will get some other enriched functional classes. Now, why I am saying this is that because this gene list that we decide is often based on the cutoffs, right. So, this is something that we choose, right, the p value cutoff or the FDR cutoff, or the if we want to impose a log to fold change cutoff, etc. So, these are hard cutoffs that are decided by us most of the time, right. So, in that case this gene list is somewhat arbitrary, right. So, this can change depending on the person who is deciding on this p value cutoffs or log to fold change cutoffs, right. So, what it means the results will be variable, right. And we are also ignoring this fact when we are doing this analysis, is that some genes are associated with multiple functional classes, and we are not considering for overlaps in functional classes, right. So, there will be overlaps in functional classes, because one gene can be part of 2 different functional classes or even more, and that information is not considered at all, right. So, we consider every gene as independent and we just do the statistical test. Now, the other approach is the overrepresentation analysis based on GSEA. Okay. So, the tool is gene set enrichment analysis tool. It is a hugely popular tool, and here is the reference and the link, right, for looking at the tool, and there is a very nice detailed discussion or the explanation of the tool. So, for GSEA, we will go into how it differs from the earlier methods, right, hypergeometric test or the binomial test. For GSEA, the input is a ranked list of genes correlated with phenotypes. Okay. So, it is not the list of differentially expressed genes anymore. Okay. It is the ranked list of all genes that are present in your assay, right. So, when you are doing this RNA-seq, let us say you have identified, you have your sequence 6000 transcripts, right. You have data for 6000 transcripts. You just rank list, you provide the ranked list of these genes or the transcripts and that are correlated with the phenotypes. So, it means you have to sort this list according to their correlation with phenotypes. Okay. And you also have phenotype labels, for example, you have control versus treatment labels, disease versus healthy labels, etc. And it actually essentially boils down to the genes that exhibit differential expression. Okay. But here you are not providing this list of differentially expressed genes only, right. What you see is that the differentially expressed genes between these two levels will come at the top, right. They will get the highest ranks because they are able to differentiate between these different phenotypes. And by the way, it is not necessary to have just 2 levels, you can have multiple levels in many cases, right. So, for example, you are doing this expression analysis for different conditions, you can have 10 conditions, 10 treatments or 100 treatments, that kind of situation. So, you can have multiple levels. Okay. So, as part of the input, you also need the gene set data, right. As I mentioned, you can get this gene set data from MSigDB or other databases, right, where you have genes that are part of specific gene sets or hallmark sets, right, that are defined. And once you have this data, this GSEA calculates something called enrichment score or ES. Okay. So, what you will do here is that it has a rank list L that has been provided by you, right. So, you are providing this rank list based on your experimental data. You have a gene set G, right, that has been obtained from MSigDB, right. So, what this method will do now, it will test whether some of these gene sets are overrepresented in the rank list, right. So, or in the whether these gene sets are overrepresented in the top part of the rank list or the bottom part of the rank list. So, why the top part? Because again this is a rank list. So, the top genes are very interesting because they are showing probably the most differential expression. So, if these gene sets are overrepresented at the top, this means these are probably associated with the differential expression and they have some important biological consequence. Okay. So, this method, as I mentioned, this test whether members of set G are primarily located in the top or in the bottom of the rank list, and that is what will give us more insights into the biology of the system. Okay. So, how does this method calculate this ES score? So, ES is calculated through traversal of the rank list L. Okay. So, this method will start from the top of the rank list, right, and it will traverse, it will take each gene, right, and you remember it has this gene set. So, what it will do, it will see what is the gene in the rank list and compare with the gene set. Okay. And if there is a match, right, if a gene in the rank list is present in the gene set, it will increase the ES value. Okay. And there is a weight, of course, it is weighted by its correlation to the phenotype, right. So, if it is at the top, right, so it will get this correlation and that will be weighted by that correlation. Okay. So, this increase is weighted by the correlation to the phenotype. Okay. And in this comparison, right, so this comparison is going on, if the score if the match is not there, right, so if a gene is not found, right, in the list, the score will decrease. Okay. So, you have these two counting going on, right. So, you are looking at the rank list, you are considering these genes from the gene set and then you are counting, right. You are seeing whether there is a match. If there is a match, you have increase in score. If there is no match, then there is a decrease in score. Okay. So, how do you calculate the final score? This final ES is the maximum deviation from 0 while traversing the list L. Okay. So, if you see some of these genes of the gene set are enriched at the top, right, you will see this score going up and then this will go down, right, and the maximum deviation from 0 is the final enrichment score. Now, once you have calculated this score, how do you actually generate the statistical significance, right. So, this is a very important part, right. You can generate any score, but you need to say whether this is statistically significant or not. Okay. So, in case of GSEA, the statistical significance is calculated based on a permutation test. Okay. So, how does it work actually? So, you can imagine, right, you have this list, rank list, right, you can draw here, right. So, you have this rank list of genes, right, rank 1, 2, etc. You have the phenotype labels, right, for example, P1, P2, P3, P4, right. So, you have these data sets, right, and once you have these phenotype labels, right. So, what you do is you permutate these phenotype labels. Okay. So, you change these phenotype labels, right, and see whether the gene set is still enriched or not. So, you calculate this enrichment score again. Okay. So, you perform this phenotype level permutation, right, if you have sufficient number of labels present, right, unless you have let us say 7, 8 or so, that is recommended by GSEA, right, you cannot do this process. Okay. So, it does not make any sense for to do this permutation if you have only let us say 2, 3 labels. Okay. So, in that case what you do is you do something called gene set permutation, right, when you do not have enough phenotype labels, is that you permutate these gene sets, right, that you are working with, keeping the size of the gene set the same. Okay. You do not change the size of the gene set, right. So, let us say in a gene set you have let us say 15 genes, you keep the size same, but you permutate the genes, okay, and then again calculate the enrichment score. So, once you have done this permutation, this actually generates something called a null distribution, and you perform this permutation test or permutation process usually 100 to 10,000 times, and 1000 iterations is recommended by GSEA, and this will generate the null distribution, okay, against which you can calculate the p value. Okay. So, you compare this enriched score, right, the enrichment score from the original data set, right, and then you compare with the null distribution and you calculate the nominal p value. Again, looking at how extreme this enrichment score, actual enrichment score is compared to the null distribution that has been generated by the permutation. Okay, so GSEA, we are again doing this multiple hypothesis testing, because you are testing each gene set separately, right, for the data set. So, you again have to do multiple hypothesis testing correction. So, this is required, and this is done through the FTR correction and Benjamini-Hegberg method. In GSEA, there is something called normalized enrichment score that is also calculated, it is called NES. So, this is actually done to account for differences in sizes of different gene sets, right. So, when you are working with these gene sets in MSecDB, you will see that not all gene sets are of same size, right. So, a gene set might have 10 genes, some gene set might have 15 genes, some gene set might have 50 genes, right. So, again to account for these differences, because the number of genes will affect the enrichment score, right, because you will find you are likely to find more matches, etc. So, to account for differences in size of these different gene sets, this NES is calculated, and this is calculated as the ratio between the actual enrichment score and the mean of enrichment score values of the permutated data sets, right. So, once you have done this permutation, let us say 1000 times, you will get this enrichment score for all those data sets, and you calculate the mean and take the ratio of the actual enrichment score versus this mean of the enrichment score that is generated from the permutated data sets. So, here are the references for this class. So, what we have seen is that functional enrichment analysis can give us insights into the actual biological processes, right. So, we can ask questions that are actually biologically relevant. For example, you can say whether certain gene is associated with certain disease, right, or whether it is helping in progression of a disease, or whether it is involved in response to a therapy or generating resistance to a therapy, right. We can also see whether a gene or pathway is associated with stress response, right. So, stress adaptation and so on. We have talked about two different approaches. The first one is hypergeometric test based enrichment and analysis, where we assume hypergeometric distribution, right. So, we are assuming a sampling without replacement scenario. So, the idea is very simple. We talked about a list of differential expressed genes. So, we get this list of genes, and we test whether certain functional classes are overrepresented or enriched in that list. Okay. So, that is one approach, and we do that through hypergeometric test. We discuss the method, etc., and how we generate the p value and do the multiple testing correction. This will give us the functional classes that are enriched in that list or underrepresented in that list. Now, the drawback of this method is that it relies on that list, right. So, this list is generated by these p value cutoffs or log fold change cutoffs, and is a bit subjective. So, depending on the person who is doing this analysis, so we might get different results for enrichment because the list might differ. Okay. In addition, this hypergeometric test does not take the overlap in gene sets into account, right. So, we have a single gene could be part of multiple functional classes, and this overlap is not taken into account when we are doing this hypergeometric test. We assumed all these gene sets are independent and we just go on do the analysis. So, there is another approach that we have talked about is the overrepresentation analysis, and the method is GSEA. Okay. So, we have talked about this, and the goal of this method is slightly different. So, it starts with the ranked list. Okay. So, ranked list of genes that and their correlation with specific phenotypes, and for we also have the phenotype labels. Okay. And what GSEA does, it looks for gene sets being overrepresented or underrepresented at the top or the bottom of this ranked list, right, because this will give us the most relevant biological information. Okay. And it calculates something called an enrichment score from this process, and through the traversal of this list it calculates this enrichment score, which is weighted by their correlation. Okay. And we have seen, right, the statistical test is done by a permutation test, where the method actually shuffles the phenotype labels when enough phenotype labels are available, or it shuffles the gene sets and it generates a nominal p value, and finally we also get a normalized enrichment score calculated. Again, for each of these methods we need to do multiple hypothesis testing correction, because we are doing statistical test for multiple functional classes. So, once we have done this, we identify a list of functional classes, right, that are overrepresented, and then we can perhaps probe these classes further, and we can see whether some of these are responsible for disease progression, for example, or maybe stress response and adaptation, and so on. So, these methods actually give us valuable biological insights into the questions that we started with, right. So, when we start this differential expression analysis with those questions, right. So, what is the difference between these, for example, disease versus healthy samples, and we want to identify at the end the molecular basis or the biological process that are actually probably driving the disease. Thank you.